Hello, my name is Sean Cordova, and I'll be talking about E. coli. The scientific name for E. coli is Escherichia coli. Obviously, the common name for E. coli is E. coli. It was first discovered in 1885 by Theodore Escherichia. He was investigating a chain of food poisonings happening around his village, where people were actually getting sick and dying, and so he actually tested the water to find it, and he looked under the microscope and he discovered E. coli. There's actually several different strains which he didn't know at that time, and nowadays there's 20 plus different strains of E. coli. So it is usually located in the intestines of warm-blooded animals. It also can be found in unsanitized areas such as sewers or even your own bathroom if it's not kept clean. But the main home of E. coli is within the intestine tract of warm-blooded animals. There they actually help to provide vitamins such as the B vitamins and K vitamins to the human body by helping absorb those nutrients while your food is going through your digestive tract. Um, his life cycle is actually pretty quick. It unlike like most bacteria, they usually replicate pretty quick. But E. coli is actually one of the faster reproducing bacteria. They usually reproduce one generation every 20 minutes, so that means every individual cell will replicate itself or reproduce asexually every 20 minutes. The average number of E. coli that's usually in a human being is usually around a billion plus cells, which most of them are shed during excrement when people use the restroom, and this quick replication helps it replenish the supply of E. coli needed for the body. Um, an interesting fact that I found that's actually used for E. coli, that E. coli is used in genetics today, is it's used for gene cloning, just because it is able geneticists are able to implement genes into the E. coli due to transformation, which means they're able to pick up naked DNA or loose DNA around them, and without it damaging their structure or their own DNA code. Because of this, and because they replicate so fast, geneticists are able to choose a specific gene and introduce it to their genome. They take it up and then replicate, and that gene is copied into the newly replicated cell, which is then can be extracted by the geneticist for study or use, or that can be tested to, I guess, diseases and whatnot. Um, and I believe that's all I really need to mention. Um, Thank you for giving me this opportunity to redo the presentation. Sorry if my volume is really low. And I'll see you next class. Bye, Professor.